Mendel performed two types of crosses. They are called as monohybrid cross and dihybrid cross. During this lesson, we will understand that what are monohybrid cross and what are dihybrid cross. Monohybrid cross describes the cross fertilization of two true breeding plants that differ in just one character. If two plants they differ in just one character, such a cross is called as monohybrid cross. Here if we take the example that there are two types of plants, one plant is producing purple color flowers and the second plant is producing white color flowers. Because we are studying monohybrid cross, so we will consider only one trait that can be purple color and white color. So such a cross in which we just consider or perform a cross by considering a single trait, such a cross is called as monohybrid cross. What was the result of monohybrid cross? So as a result of monohybrid cross, Mendel determined the segregation of alleles at a single gene locus. Now if we see what is the hypothesis behind the dihybrid cross, in case of dihybrid cross that determined alleles at two different loci, what kind of is the segregation? Whether alleles at two different gene loci they segregate as a unit or they segregate independently from each other. The experiment can be of seed shape. Seed shape is controlled by one gene pair, while the seed color is controlled by another gene pair. Mendel crossed pure breeding plants which were producing a round and yellow plants seeds, while the second type of the plants they were producing green and wrinkle seeds. As a result, if there is a dependent segregation, then both the alleles they should segregate as a unit. They should transfer from the parents to the offsprings as a unit. They should not segregate independently. In case of dependent segregation, like here is the example of dependent segregation. Parental plants, one of the parental plant that have gametes capital R and capital Y, while the second plant that have gametes small r and small y. When F1 generation will be produced, that will have capital R, capital Y, means one of the gamete from the father, while small r and small y, one of the gamete from the second plant. If they are joined together, if there is dependent segregation, then same kind of the alleles will be produced during the result of the plants of F1 generation. So the gametes of the parents they will be joined, and the gametes of the plants of F1 they will also be joined. So the segregation will be, the ratio of the plants will be 3:1. 3 plants will be producing a round and yellow seed, while one of the plant will be producing a wrinkle and green seeds, if there is a dependent segregation. While in case of independent segregation states that the gene loci at two different positions they will segregate independently. They will not inherit as a unit. If this is the case, then if the segregation is independent, then two types of the gametes will be exactly like the gametes of the parents, while two new types of the gametes will be produced as a result of independent assortment. If we take the example that this is the gamete like the parental gametes, this is the gamete that is also like the parental gametes, but there are two new types of the gametes will be produced during the process of independent assortment. So in case of independent assortment, characters which are transmitted from the parents to the offsprings, question arises whether they are transferred as a package, as a unit, or they are transferred independently. The answer to this question is that they transfer to the next generation, to the offsprings, independently to each other. So this is independent assortment or independent segregation. Here we can take the example of dependent segregation and independent segregation. If there is dependent segregation, then the ratio should be produced 3:1, like in case this one. But in case of dihybrid cross, this ratio was not produced. Instead, the ratio was produced, that was 9, 9, 3, 3 and 1. So 9:3:1 ratio indicates that the segregation was independent to each other. So if we conclude monohybrid cross and dihybrid cross, monohybrid cross is the cross fertilization of the plants that differ in just one character, like purple color and white color of the flowers. While in case of dihybrid cross is the cross fertilization of the plants that differ in two different characters,